Thank you for joining us for the quarterly lecture series for the Network for Ocean Worlds. NOW is a NASA astrobiology program funded research coordination network that links researchers studying the oceans of Earth with those studying the oceans of planetary bodies. In this first season, we're looking at life on ocean worlds. Our first quarterly episode was an introduction to cryospheres, both here and beyond. Our second episode looked at life on seafloors and in oceans. And in this third episode, we're going to be looking at life in the ice and at the ice-ocean interface of both Earth and ocean worlds. To talk about Earth, we invite Dr. Kevin Arrigo from Stanford University, while Dr. Michael Malaska from NASA's Jet Propulsion Laboratory will tell us about planetary oceans. These talks are about 20 minutes each, with discussion following. Thank you very much for joining us.
So for those of you who don't know me, and actually I'm gratified to see so many familiar faces under the participant list. My name is Kevin Arrigo. I'm at Stanford, as Sam said. I actually been doing sea ice work for a long time. I did my PhD on Antarctic fast ice ecosystems a long time ago, and I've been doing some sea ice work ever since, although I have sort of broadened my interest into other areas. So it's really impossible to understand life in ice without understanding something about ice itself. And so I'm going to begin with just talking a little bit about ice structure and ice dynamics and how that relates to the issues of how things live in ice. So this is a sort of a typical cross-section of an ice flow. You could find this in either the Antarctic or the Arctic, it doesn't really matter, although fragile ice is more common in the Antarctic. What you see here is ice that's layered. There's a surface fragile ice layer, there's an internal columnar ice layer, and then there's a skeletal layer at the bottom. That skeletal layer is really important because that is the growing interface of the ice. Fragile ice is it's sort of slushy ice that forms when it's kind of in the fall when the ice is just beginning to form, the air temperatures are just beginning to get cold, the wind is blowing really rapidly, and it's churning the water, and it's causing ice crystals to form in the water. Eventually, the wind stops, those ice crystals rise to the surface, and you get this layer of fragile ice. So sometimes fragile ice is there, sometimes it's not. Sometimes the ice is just columnar ice with a skeletal layer at the bottom when it's growing. To move this so I can see. So on the other side of the screen, it shows where the different communities are. And so it's sort of where you can expect to find microbial life. And most of the life in the ice is microbial life, although occasionally you find things moving into the ice to eat that microbial community. Almost virtually everything I'm going to talk about today is microbial. So sometimes you find it at the surface, although those tend to be relatively rare. Internal communities are a little bit more common, and the internal communities typically form in ice that's rafted. So you could have one flow that's sort of rafted on top of another flow, and at that interface you may get an internal community form. But much more common what you find is the bottom community that lives within the skeletal layer. And occasionally you'll see a strand community. It's sort of a community that hangs down from the bottom. And I'll show you pictures of all these different communities in a little bit. All right, so this is a close-up now of the skeletal layer. So sea ice is really different from other types of ice. It's different from glacial ice, it's different from lake ice, and the reason it's different is because it's growing in a salty medium. And because it's growing in a salty medium, this is the growing ice crystal structure, and these ice crystals extend vertically downward into the water as the sea ice grows, as it freezes. When it does that, you can see it sometimes traps little areas here of seawater, and those are called brine pockets. And so as these brine pockets get incorporated in the ice, it gives the ice salinity. So the ice becomes salty, certainly saltier than you would find in the fresh water or glacial ice, for example. And the consequences of that salt is that salt wants to migrate back out of the ice again. And there's a variety of mechanisms for doing that, and I'll talk about those in a little bit. But the key thing here is sea ice is salty ice. So this is a plot showing typical cross-section or typical profiles through ice of different thicknesses. This is actually ice followed over time. And so on the right side is temperature profiles. And so this would be sort of an early fall, relatively thin piece of ice, and this is the temperature profile. And then as you move from fall to winter, the ice gets thicker, the ice also gets colder. But you'll notice that the interface with the ocean, which is the bottom of the ice, it's always the same temperature. It's always at the freezing point of seawater. Higher up in the ice, though, as you get farther and farther away from the water, it can get colder and colder. And in fact, it can get really cold, much colder than I've shown here. It really depends on what atmospheric temperatures are. And surface sea ice temperatures of -20 degrees are not uncommon at all. This, what I'm showing on the left side, is the bulk salinity. So it is the consequence of those brine pockets being trapped in the ice. And the bulk salinity averages about 5, which is about 5 parts per thousand. We don't use units in salinity anymore in oceanography, but I'll use them here. So it's about 5 parts per thousand. And so ice is relatively salty compared to what you would find in freshwater or glacial ice. So that was the bulk salinity, though. In terms of the microbes don't live in ice crystals that are not associated with brine. So if there's any microbial life in the sea ice, up in the ice where the brine pockets are, they're going to be living within those brine pockets. And those brine pockets have characteristic salinities that are determined completely by the temperature. So the colder the temperature, the higher the brine salinity at thermodynamic equilibrium. And so if you're going to have liquid water in the ice at 25 degrees below zero, it's going to be about 225 parts per thousand. It's going to be really salty. And so the brine salinity of those brine pockets up in the ice can be really, really high. And that is actually what limits where microbial life can live in the ice. It's not the temperature so much. They can grow pretty well at low temperatures, but they have a really hard time growing at these very, very high salinities. Another important characteristic of the ice is the brine volume. This is done in terms of brine volume fraction, where 0.5 would be basically that. 0.5 would mean 50% of the volume of the ice is taken up by those brine pockets, which would be really high. You almost never find that. But what that tells us is how much of the ice is actually available for microbial life to inhabit. Not only that, brine volume fraction is really important in terms of defining the connectivity of the ice. Whether or not brine channels form, how easy it is to get salt out of the ice. The higher the brine volume fraction, the more salt goes out of the ice, and the larger area you've got for microbial life to live. Now, what determines the brine volume is two things. It's determined by the temperature, and it's also determined by the bulk salinity. So with temperatures warm, more of the ice becomes brine. As the ice becomes more salty, more of the ice becomes brine. And so that's important in terms of, at this point right here, at about 5 parts per thousand, which is 0.05 fraction, about 5 parts per thousand. This is when those brine pockets get big enough where they actually start to merge and they connect together. And the more connectivity, the easier it is for microbial life to move in and for things like salts and other solutes to move in and out of the ice. So this is just a close-up picture of some ice crystal surfaces. The one on the left side here, this would be cold ice or relatively fresh ice, and you can see it's got brine pockets, but the brine pockets are all pretty separated, right? And so it would really be difficult to move things in and out of the ice when the brine pockets look like this. But when you warm the ice, what you do, or you have saltier ice, what you can find is that there's a lot more connectivity, and you can get a lot more things in the ice and out of the ice, which is a lot more potential for microbial life to live within that ice.
So Kevin, while we're on the slide, we had a question come in on what's the average geometric size of these brine pockets and how much do they vary?
Well, they're like millimeter scale or smaller.
Thanks.
Okay, this is just This is X-ray microtomography of a 3D version of an ice crystal, showing the connectivity now and the coalescence of a bunch of brine pockets. And so, as I said, this makes it a lot more feasible for things to move in and out of the ice and microbes to move in and out of the ice. So this then so one of the consequences of that is that the salinity of the ice changes over time. So this is a time series. So this is a cross-section of a piece of ice, so from the surface to the bottom, over an entire season. And it's really hard to tell from this, but what's happening is you're losing salt. You're losing salt out the bottom, and so it tends to be really salty at the bottom. Sometimes the way the physics of the ice works, it also pushes ice up to the surface, or not ice, salt up to the surface. And what you often find is relatively salty surface ice as well. But generally what's happening during this time is the ice is desalinating, as ice is getting older and older and getting more and more porous as temperatures begin to warm up. So at this stage here, you'd expect to be really losing a lot of salt, but you can see there's a lot less salt at the end here of the time series than there is at the beginning. And so what drives that is brine drainage. So what we've got is we've got these brine pockets that are connecting into brine channels. Dense brine is flowing out from here. And the important thing is, as that dense brine flows out, it is replaced by seawater that is fresher and, more importantly, also high in nutrients, which is going to be really important for those microbes. So nutrients are moving in and out as the salt moves in and out. So this is brine drainage. Now this isn't as important as the next process, which is skeletal layer convection. So this is that skeletal layer I showed you before. As these ice crystals are growing downward into the water column, what they do is they exclude salt. Right now, some gets trapped, but most of the salt is actually excluded. If all the salt was incorporated, sea ice would be the same salinity as seawater, and it's not. So a lot of brine is being excluded. As that brine flows out from in between these little tiny fingers, and again, this is sub-millimeter scale. This is really tiny. What that does is also produces convection, which brings more nutrients into that skeletal layer, a really important process. So if we look at a sea ice environment as a whole, when the surface of the ice is colder, the brine salinity tends to be high, the brine volume is low, brine channels are not very well developed, and there's little exchange with the ice. And this is less favorable for life under these conditions. But at the bottom, when that skeletal layer is actively growing, it's convecting, that area is actually more favorable for life when the ice is cold. But later as the spring and summer move on, brine salinity drops, brine volume increases, the brine channels become much more well developed, and there's much more exchange within the ice, which makes things in the interior of the ice more favorable for life. Things are still okay at the bottom, but they're not quite as good because you need that skeletal layer convection to keep moving nutrients into the bottom of the ice to allow the microbial community to grow. So let's just look at some of those communities. So this is a typical surface community. It would probably be between you can see where there's ice and snow underneath. It's probably laying right at that interface. They typically, you almost never find them right on top of the ice because it's just way too bright. This is an interior community, and this one, in fact, has formed at the interface between two rafted ice flows. One ice flow that sort of got pushed on top of another one, and there's a community right in the middle, right there. This is a bottom community from underneath the ice, and you can see it's relatively dense. And you can see that really clearly when you actually do a core through this ice. This is the bottom community in the core. So it's not very thick. This is only maybe, well, here it shows. This is maybe 3-4 inches, but it's a really dense algal community in that 3 to 4 inches, and that skeletal layer. So this is where the skeletal layer is. And you can tell where the boundary of the skeletal layer is really clearly because there's very little microbial growth above that. And finally, this is that strand community. These are not that common, although you do find them sometimes in the Arctic and sometimes in the Antarctic. They're more common in the Arctic than they are in the Antarctic. Central Arctic is really common for these. Okay, so the inhabitants. it's dominated by diatoms. Diatoms are single-celled photosynthetic microbes, I'll call them, that have a silica shell. And these things are then preyed upon by a whole variety of ciliate species, which are the most dominant heterotrophs. But these are the dominant food-producing organisms in the ice, as the diatoms. So they're photosynthesizing, making their own food, providing food for the rest of the sea ice ecosystem. So diatoms are key. There's other things there, but they're not nearly as abundant as diatoms. And the reasons are as follows. Diatoms have a lot of adaptations that make them really good in ice. One is that they are actually adapted to glide along ice crystal surfaces. Not many things can do that. You need to be able to maintain your position in the ice if you don't want to get frozen in while the ice is growing, right? You've got to move along with that growing ice interface. And so you've got to be able to glide along ice crystal surfaces. And to do that, you have to stick. And so they use carbohydrates and protein-based, it's not really glue, but sort of like glue, that allow themselves to stay attached to the ice, whereas a lot of other microbes can't do that. They are adapted to a wide range of salinities. They do best around seawater salinity, but they can survive, they can grow at about half their maximum growth rate at salinities ranging from about 10 to up to 60 parts per thousand, which is a pretty wide range. And even they can grow, they can continue to grow at up to almost 100 parts per thousand, which is very salty water, or brine, I guess in this case. Because they have to deal with salt, they're really good at osmoregulation. And so they produce a variety of osmolytes. One of the most important ones is DMSP, which is this molecule here. But they also produce another molecule called glycine betaine, and there's others as well, that they use for osmoregulation. And it's this that allows them to survive in those really high salinities that you find in the interior of the ice, as well as the low salinities when you've got meltwater flushing through the ice, which happens occasionally. Although the high salinities are definitely the bigger problem than the low salinities would be. A key thing for them is that they're really good at growing at very low light, right? Underneath the ice, especially if there's a snow cover on top, there's very little light getting through that ice. And so they have adapted to have a very high photosynthetic pigment content. That's why you saw that layer in that core was so brown, they're just packed with pigments. They've got a very high photosynthetic efficiency, which is what this shows. Their growth rate increases really quickly at very low light levels. So net growth possible only 1 micromole photons per meter squared per second. To put that in context, on a sunny day at noon, it would be about 2,000 micromole photons per meter squared per second. So this is a very low light level.
So Kevin?
Yeah.
Just looking for a break. We got a question in about whether you can discuss some of the difference between Arctic and Antarctic sea ice, particularly seasonal ices.
The main differences are Antarctic ice has more fragile ice. Arctic ice doesn't have nearly much at all because Arctic it's a much more windy, open ocean environment. Arctic is more protected. The other big difference is the Antarctic has got a lot thicker snow cover on it than the Arctic tends to. And you get a lot more melt ponds that tend to form on Arctic ice than you do in Antarctic ice. But in terms of the main ice structure, it's the same. The congelation ice part, where it's just your typical ice crystal formation, it's the same. Okay, so they're also adapted to very high light levels, which occasionally happens to these guys. If they had a really heavy snow cover on top of them, suddenly that snow cover is gone. They've got to deal with high UV radiation. And so they've got these sunscreens where they absorb very strongly in the ultraviolet region, which is protecting them from too much ultraviolet as well as visible radiation. They can also produce antifreeze. So this is a stained, where it's this transparent exopolymeric substances that they produce that inhibit ice crystal formation and protect cell membranes from freezing. And so this is a diatom. There's another diatom. This is a brine pocket right there. And so that brine pocket is making sure, that diatom is making sure that that brine pocket does not freeze and kill that cell. And they can also regulate membrane fluidity, which is really important. At cold temperatures, maintaining membrane fluidity is important for a lot of metabolic processes, particularly photosynthesis. And so it produces a lot of this MgDG, which is a fatty acid, which as you increase the number of double bonds, it makes it more fluid, and therefore it increases electron flow, for example, which you need to maximize if you're going to be photosynthesizing efficiently at cold temperatures. So the whole variety of adaptations they've done to deal with this really harsh environment that they live in. But there are other things that live there as well, bacteria, and I'm not going to go through all of them because there's thousands of them, but what I would talk about is there's a lot of different metabolisms that...
So, on the subject of metabolism, before we move on too much, what's the maximum ice thickness that still allows enough light for photosynthesis?
It depends on snow. So if there's not snow, you can get easily enough light through 2 meters of ice with no problem. Even a little bit of snow cover, though, reduces that dramatically. The amount of light attenuated by snow for a given thickness is 10 times more than that of ice, which is about 10 times more than that of water. So snow is the key. Ice thickness isn't the key, it's snow is the key. So back to our bacteria, there's a lot of different metabolisms. There's heterotrophic bacteria that basically break down organic matter into its constituent parts, and primarily ammonium in terms of nitrogen, and all these are, it's a very nitrogen-focused slide here. Nitrification, which is a type of chemoautotrophy where they take ammonium and they extract the energy from that ammonium and fix carbon into organic matter. There's been measured denitrification. Sometimes those brine pockets become anoxic, and when those brine pockets become anoxic, you cannot undergo standard heterotrophy, and so the electron acceptor in this case ends up being nitrate or nitrite to oxidize that organic matter, and you produce N2 gas. And even anammox, which is a relatively recently discovered nitrogen metabolism, it's a form of chemoautotrophy where they use nitrate to oxidize that ammonium that's produced during ammonification for growth and to fix carbon. And again, this produces nitrogen gas as a byproduct. So lots of different things going on within the ice. So the last thing I was asked to do was to talk about the application of sea ice to other bodies. And so I think hopefully I've given you a feeling for how microbes, they've adapted to a wide range of environmental conditions, but not finite, right? They can't deal with, if salinity gets too high, they can't grow. They can live at very low radiances, but, they need some light, right? And so they tend to grow throughout the ice, but they tend to do best where conditions are mild. At the bottom is where you find most of them. You'll find them in the interior, but they're never doing as well as they are at the bottom of the ice. The key, though, for ice algae, though, is that it requires a dynamic ice flow. The ice has got to be growing. If that ice is not growing, those convective processes that are bringing new nutrients into the ice shut down, and all you'll have is a very thin layer of cells maybe growing at the undersurface. You won't have those dense communities that I showed you in some of the slides. And finally, photoautotrophy is dominant metabolism in sea ice, but chemoautotrophy is also present, so it's also possible. And with that, I think I will stop. Thank you.
Wonderful, Kevin. Thanks so much.
Mm-hmm.
I absolutely learned something during that. So, right, which is the point. It's nice to get some of the terrestrial perspective and ground ourselves a little. So we have a little bit of time before we're going to move on to Mike Malaska's talk. And if there are people who'd like to ask their questions over audio, you can feel free to unmute and do that, or continue asking them in the chat, and I can put them here.
Hi, this is Robert Drucker. I have a question. Are you hearing it?
Go for it.
Okay. So you just said that active microbial community requires dynamic growing ice conditions. So, so does that mean that this only occurs in the fall when there's still light, and there's still some light, rather than during the melt season when the light returns, but the ice is melting rather than growing?
No. So active year is sort of a relative period. I mean, you will have a microbial meeting. You can have it any time, but sometimes you get them in the fall. But you can also have them in the early spring when the temperatures are still cold and the ice is still growing and there's still plenty of light to grow. That's when you get most of the activity in the algal community there. It's going to be in the springtime before the ice begins to melt. It's still freezing. It's not growing as fast as it was, but it's growing fast enough that convection is still happening and bringing new nutrients in and providing nutrients to that dense community that you can do. In the fall, you get a little bit of a buildup, but it doesn't last long enough before the lights go out and things shut down.
Thanks. So we have quite a few questions coming up in the chat. Natalie Wolfenbarger mentioned that likely permeability is super important. So how much does the columnar structure matter in governing the permeability threshold? So and then would you expect a difference between columnar and frazzle ice at the same temperature?
Oh yeah, that's good. I'd actually mention so frazzle ice is more permeable, and so it would tend to support a more robust community than congelation ice or the columnar ice. Columnar ice is, particularly as it ages, it gets less and less salty. There's less and less brine pockets, and so there's less places for microbial communities to grow as the ice gets older throughout the winter, right? And so by springtime, it's gone from maybe 10, 15 parts per thousand down to maybe 5 parts per thousand. So it's not nearly as good a community. But if there's frazzle ice, that tends to still be pretty salty and a lot of brine volume there for things to grow.
Great. We have lots of really good questions coming in. So is the structure of the ice and microbial communities in sea ice very different from what we would see in hypersaline lakes inland?
Oh, well, yes. I'm trying to think of if any hypersaline lakes freeze. And so, I mean, without the ice component, it's hard to compare them. The salinities are comparable, though. Salinities in ice, as I said, can be 250 parts per thousand, which is really salty. I'm not sure maybe some hypersaline lakes get higher than that, but that's almost 10 times seawater salinity. And nothing we've seen can grow there. Now, the algae we're looking at are algae that were probably adapted to grow in seawater initially, and they've moved into the ice, and that's why they still do best at seawater salinities, although they can grow at, like I said, almost 100 parts per thousand, which is 3 times seawater salinity. And so I think it's comparable, depending on how hypersaline those lakes get, and I'm not sure about that.
Great. So we will get to the rest of these questions as they're coming in. Now I think we should switch to Mike Malaska, and then at the end of both talks, we'll be able to come back and have some more discussion.
I'll introduce myself a little bit. So I used to actually have a career in the pharmaceutical industry. I'm trained as a synthetic organic chemist. And I recently joined Planetary Science about 7 years ago, and then I have recently become more and more interested in the astrobiology of the ocean world. So I'm still relatively new to this field. What I want to talk to you all about today is life in the deep ice, both in terms of Earth and also for the ocean world. So I'm going to basically be riffing off the stuff that Kevin already presented. And I'm going to set the stage a little bit and talk about the environment that's way down deep in the crust and in the Earth's ice sheets. And that's an area where you have no sunlight. So you really don't have any you're way beyond that 2 meter depth. You're under high pressure. It's very low temperature. And as Kevin had mentioned, you have these little tiny physical microenvironments of these little tiny brine pockets where you also have a lot of good chemicals concentrated up that could be nutrients and building blocks. So I'm going to just cut straight to the coolo chase and show you a picture of a microbe in sea ice that's in a brine pocket. So this is from Karen, who's I just saw in the audience as well. So this is some of her work. And in this brine pocket, you can see these little tiny microchannels, and you see a triple junction between 3 separate ice grains. And if you zoom in, you can see this little tiny pocket, this little place that's unfrozen because of this high salt concentration. And if you light it up with a fluorescent stain, as you see in the lower right panel, you can see that there are two little tiny bacteria that are kind of hanging out in that tiny little pocket. Okay, so as Kevin was presenting on ice, as ice forms, you have this nucleation of the ice, you have pure water forming a beautiful crystal, and as it crystallizes, it freezes and basically pushes out all of the chemical impurities. So at the end stage, you have all of these pure ice grains, and they're all surrounded by all these little impurities that as you increase the salt concentration, it will decrease the freezing point. So as long as you don't go too cold, you will eventually have these tiny little concentrated salt zones that remain liquid, depending on your local temperature. And this is where you would find that beautiful concentration, a beautiful environment for these microbes. Okay, so somebody did a cool experiment, and they took ice. They did this as laboratory ice, and they've also investigated it with Greenland ice from about 146 meters down. So this is the top of the Greenland ice sheet, about right in dead center of Greenland. And they took some of this ice, and this is ice that came from snow, pure snowfall coming down. So not really seawater, but it's actually fresh water and what you get out of the clouds, and then compacts down to form ice. And they zapped it with a UV or a Raman system, and they measured the salt concentrations that are actually in the different areas. And what they found is when they zapped this little tiny triple junction micropocket, they found really high concentrations of sulfate and nitrate, up to about 200,000 times higher. So as Kevin was mentioning, you can have some of these metabolisms that are using some of these materials. Well, here you have this incredibly high concentration that's being done for you by the ice forming. So you have these great little pockets that are just chock full of nutrients and materials, and these little bacteria can kind of hang out there and live with all this concentrated food supply. The trick, though, is unlike for sea ice, where you can actually have fairly high salinities to start with, here we're dealing with pretty much pure snow. So your volumes are really tiny. So you maybe have 100 microliters of volume for every liter of bulk ice. So it's a tiny little pocket that's very far separated from each other. Okay, there's another trick, and that as these ice grains grow, they tend to push out stuff. And it actually ends up being size dependent. So the smaller your chunk, your particle, the better you are at being able to surf that wave and stay in that little brine pocket. If you're too big, you're basically going to get encapsulated and embedded in the ice. And Kevin talked to you about these diatoms that are able to actually excrete materials that help them kind of scoot along and survive being stuck in the ice. For other bacteria that don't have those cool little tricks, they effectively have to be small. And that's actually going to end up driving the sizes of bacteria that you're going to see in the ice. They got to be small little guys, or they're going to get stuck in the ice. So here's sort of like a quick graphic that shows some of the different sizes of cells that we see out there. So red blood cell is a pretty big thing, about 8 microns. Bacteria, E. coli, it's about 1.5. Staph A, 0.9, Mycoplasma genitalium, which is a sexually transmitted disease that we'll be hearing, unfortunately, a little bit more about. That's fairly small. That's also the bacteria that has about the smallest genetic component to it. I think it's like 350 genes which are expressed, which is about the minimum set that you need to have a viable microbe, free-living microbe. The deep ice microbes that they've found are about the order of 0.2 microns. So they're really tiny. And in fact, we can look at it in the upper right corner here. There's a typical filter, frit, of about 0.2 micron pores. And you can see 2 little bacteria that are basically kissing each other and not going down through that little hole. But if those bacteria were separate, they go straight down. So that's also a good thing for us to think about when we're going into nutrient-starved and cold environments, that we may need to have filtration systems that are a little bit smaller, a little bit finer than what we had originally used.
So Mike, As you started talking about our organisms and their environment, Robert Drucker asks if sea ice also absorbs atmospheric oxygen or other gases. So in addition to what's coming down into the snow and being trapped, is there also gas exchange going on through the sea ice layer?
It gets wilder than that. I can talk about that at great length, but there's one cool little thing is that you can actually have your ice, as you get down to a certain depth, you're going to hit the point where you actually have clathrate stability zone on Earth. That's about 1 kilometer down. And your ice itself will actually start encapsulating oxygen and nitrogen and changing the local gas environment that you have. So when you're thinking about deep ice, that's actually something important to realize is that the environment is going to be dependent on the physical conditions of the ice and the chemical structure that it's going to want to have. Hope that answers that question a little bit.
Thanks.
Okay. Next slide shows sort of like a rogues gallery of several bacteria that have been isolated from the GISP2 core down about 3 kilometers deep. And I'm going to note the pressure here. That is the pressure here that is about 30 megapascals and 264 Kelvin. So this is starting to get conditions that we would see in some of the icy crusts of the ocean worlds. That's kind of a cool thing to think about, is that these are bacteria that might be under similar conditions. And you'll notice that they're all fairly small, and there's a lot of different little guys that are down at that zone. Showing the next slide. Uh-oh. Did I just do something bad here? Righty. Uh-oh. I may have done something bad, guys.
I see lots of gray boxes popping up on your screen, Mike.
Yeah, I'm gonna kick out of that. Sorry, I think I allowed somebody to annotate it, and I don't think that was an okay thing to do. So sorry, guys.
Yeah, sorry, Mike. I hit a button on my WebEx by accident, and somehow it impacted your presentation. Never seen that before.
And I just said, sure, Alexis is cool. Let her have fun with it. Okay, cruising on. So this is a transect of the GISP2 core. Over here on the far left is a section. This was drilled at Summit Station, Greenland, which I'll be talking about a little bit later. But it's about a 3 kilometer deep core. They went all the way down about 50 centimeters into the base of the dirt, effectively, at the bottom of the core. And they sampled all the way through and measured cell densities. And about 1 kilometer down, you're at about 1 to the 5 cells. You get down a little bit deeper, you're tending to places where the ice is a little bit warmer because you're closer to the geotherm of the Earth. And this is a point where microbes can actually start to live. There's a magic number of about 250 Kelvin, which is about the cutoff for when cells can either survive and metabolize, or they're basically frozen. And we'll talk about why that is in a few seconds. But the cell densities are around 1 to the 6, and as you get way down deeper, you get to a point where you're really close to the Earth, warm geotherm, you have salt flushing nutrients through there. You can have really high cell concentrations, about 1 to the 8. I have made a quick chart showing you relative cell concentration. So the absolute theoretical limit that you can have, assuming a volume of a cell, is about 1 to the 15. On the other end of it, medically sterile material is about 0.1 cells per cubic centimeter. And in ice, glacial ices, we see anywhere between 1 to the 2 to 1 to the 8. For ocean worlds, we might be in a more nutrient-starved environment. So in that case, we may be anywhere between having 1 to the 6 to all the way down to basically sterile ice. So our instrumentation should be set to be able to detect materials at those concentrations. Right. So as they examined the GISP2 core, they actually found an incredible diversity down there, a lot of different phyla that are present in the ice. And more fascinating is even around 2.5 kilometers down, which is above the point where your cell is going to be able to metabolize, so active metabolism, and high pressures, about 25 to 30 megapascal, even under very small vertical changes of depth, you actually see large shifts in the population. So that's telling you that there's actually a very diverse environment down there. And that suggests that you could have tiny little microenvironments from one place to the other, and they could be very different from either one depositional period or from one chemical environment to the other, which is really fascinating. All right, so a quick thing, and this is something that Kevin touched on as well, is what is it that makes cells die? What's your cutoff? And it turns out that it's the interior of the cell undergoes a glass transition right about 250 Kelvin. So it basically freezes shut. That's okay because effectively your cell activity stops. But what's bad is if your cell activity doesn't stop, but your outer membrane freezes. In that case, you don't have exchange of food across the membrane. You have bad things that can occur, such as freezing from outside inside or freezing from inside to outside. You rupture your membrane, and all these things are very, very bad things to have happen. So cells have adapted a various number of strategies to be able to deal with this. And in terms of general concepts, these might be things that ocean world organisms might do as well, because they're faced with the same problems. And some of the solutions might be things that we could look for in terms of a potential detectable biosignature that we could measure. So a very quick, brief run through of dealing with cell membranes. Basically having a membrane go to gel stage is bad. So cells have developed a lot of little clever tricks to be able to modify their membrane, either in situ or during synthesis. And those might be things that we would detect, or we should be prepared to detect. Kevin mentioned polyunsaturation. There's other little tricks like putting in cis double bonds as well. I have way in the deep backup slides a really cool slide that describes how these cells actually detect this and then can modify their cells. But just for thinking of potential biosignatures, all of these things are potential things that we might want to look for when we're considering fatty acid patterns, for example. Another thing, and Kevin also touched on this, is that these cells have abilities to control ice. So they can set up proteins that have hydroxy groups that sit out there, and they actually set up an ice structure, and they can either form ice or they can prevent ice from forming. And one of the really cool things I'll just throw out here is that it turns out that as these cells, these proteins create an ice structure, it's not actually ice-1H that they form at the protein interface, it's actually a clathrate. There's actually a little pentagonal structure that is set up, and the ice lines up in that form as a partial clathrate face, and then from there forward, it becomes regular structure 1 ice, which is kind of cool. Commercially, this is very interesting because you might be able to create ice-binding proteins that can deal with commercial produce, freezing damage, and also one of the things I heard is that it might actually end up making a better ice cream, which is just really a neat goal right there. Okay, so now moving to the ocean worlds, thinking about the different environments on out in the solar system, focusing more on the larger icy crust, deep subsurface ocean bodies. So we have Europa, Ganymede, Callisto, Titan, and then I've thrown Triton in here as well, but we can also throw in Pluto and some of the other places. Okay, so looking at those bodies, Earth and Mars both have perched oceans, so 3-kilometer thick precipitation-driven ice sheet. Enceladus, Europa, and Titan, Ganymede and Callisto, all have oceans that formed from freezing. They also have a deep subsurface ocean, so you have freezing from below, as Kevin mentioned during ice formation. For the larger bodies, you have convection in the ice, and that will allow you to maintain a warmer temperature throughout the entire icy crust. So it's not that super cold that you have on the top surface. Down deep, you have a little bit warmer ice where you might start approaching some of these temperatures that you see in the Greenland ice sheet. So as Kevin mentioned, at these ice-ocean interfaces, you have these ice zones that progress inwards. They trap some of the near-surface materials, brines, salts, they dewater. You eventually will entomb some of these materials in there. And then you can actually think that it's possible that as freezing progresses, you start jacking up the concentration, and you may start getting convective transport. You'll have some possible chemistry happening. You can have this really neat mixing and exchange of these different microenvironments. you could have, even though it looks like boring ice and it all formed from the same stuff, you could have a whole series of wild microenvironments that exist throughout the ice. And I'll throw out here just an idea that it's possible that some of this chemical concentration driving mechanisms from ice freezing may have actually caused or create conditions for the formation of life, the origin of life. And I'll reference some of T. Buford Price's work on that. But effectively, you have a very rich possible variety of microenvironments in the ocean crusts of these ocean worlds. Okay, next slide talks about some of the temperature conditions that as you go down through Europa, Enceladus, and Titan, you get around 10 kilometers, 7 kilometers deep, and now you're starting to cross that threshold where cellular metabolism can occur. So you could have a living microenvironment habitat in the ice. You are under a little bit of pressure, and that may not be that bad of a problem. So the next slide shows the pressure and temperature conditions of Europa. And I'm going to introduce several different bacteria that can live under some of these conditions. So Planococcus halocryophilus, I mean, just from the name, you know, high salt, low temperature, you're at negative 15 Celsius, 258 Kelvin, and it's able to grow and metabolize just fine. You have these other ones that have been isolated from deep in the GISP2 core, and they can handle high pressures and low temperatures just fine as well. Cycromonas has been found in the deep ocean. It can do high or low temperatures and high pressures as well, just fine. So effectively, looking at this and looking at the Europa traces of expected conditions, you can see that we are in a habitable environment in a lot of these ocean worlds. So our question is no longer going to be, is it habitable, a habitable environment? The answer to that is yes. The question now is it actually inhabited? Are there actually organisms that live down there? And so that's what's going to be focusing a lot of our future exploration, is the actual search for life. So I've drawn out kind of a schema here. Europa Clipper mission, which is, you know, slated to launch. Lander, which is still mission concept phase, and then finally, far future, you're going to have some sort of a deep drilling system, which is going to go through the ice crust and then actually go to the ocean. So recalling that the deep ice is going to be your first habitable environment that you're going to get to when you're going for that ocean. You're going to want to have the tools and instrumentation that is going to be able to examine microbes or potential microhabitats as you go down through the ice. And so this slide shows some of the potential, kind of like the state of the art for some of these systems. You have a drill mobility system, which would be at the front end. We have some sort of a detector that says, hey, there's something interesting through the ice, we should sample acquisition system, detailed analysis system, and then finally at the back end of it is you want to have some sort of an explorer to bop around and look for things in the ocean. So one of the things I wanted to focus on is some of our work that was looking at an initial detector system. And what we decided to use for that was a deep UV spectrometer that would be able to basically shoot a laser, ultraviolet laser into the ice, and then detect if anything aromatic fluoresced and glowed back at us. This is the same type of technology that has been recently launched and is on the way to Mars as the Sherlock instrument on Mars 2020 Perseverance rover. And so we basically readapted it for ice. Yes.
Mike, you have a question? Okay, never mind. It was clarified. Continue.
Okay. Cool. All right. So next slide shows some of the work that we did at Greenland. And we were sending it down to the ice. We were blasting the sides, and we created a map of information from about 93 meters depth in the ice. And we're able to isolate and detect some hotspots that exist, which is a big surprise right there. And we were also able to extract the spectra out of it so we could compare it with some of the known materials. And what we found is we got a whole series of, oh, it actually has sound. Okay, cool. You can actually see us raising the drill instrument system out of the ice borehole. So that was its last rise from 100 meters deep in the ice. We saw a whole variety of different spectral signatures down there. So we saw a lot of different things. We saw microbes, amino acids, lignans, two-ring aromatics, all sorts of stuff. So we did see a wide variety of things. One of the things that is a big lesson out of all of this is that we created these maps, like I just showed, and we saw that ice is not uniform. So there are things that are in there, microbes, organics, and they're not uniformly distributed or even in layers as we had originally thought. Instead, it appears that life likes to clump, as we kind of all know, and it continues in the ice, and that there are tiny little discrete hotspots that seem to be spectrally pure, which is interesting, and that they're not uniformly distributed, they're in little spots. And the key is that if you are going to be looking for materials, organics, and microbes in the ice column as you go into the ocean worlds, and if the concentrating behaviors and transport behaviors are similar to those on the ice sheets that we've examined on Earth, there's a very good chance that you could end up sampling something blindly and you won't see anything. You may be in one of these void spots. So that doubles down and tells us that we need to have a way of being able to detect some of these things in the ice and be able to say, hey, here's something that's very interesting, and then extract that out of the ice and then analyze it. If we do just bulk melting, we may end up going below our instrument detection thresholds, especially if when you're in a nutrient poor environment where you may have very low cell counts to begin with.
So talking about the stability, you have a question on, you mentioned the stability of these life forms at potentially high pressures and low temperatures, but what about their stability over ranges, like to show that they can survive fluctuations in these environments?
Oh, yeah. So some do and some do not. So I'm trying to remember, I believe the, I forgot the name of the guy now, the Keikoa, the isolated deep ocean microbe from Nori et al. I believe that was an obligate piezophile. So you had to culture it under pressure or it would just not be happy. There are some organisms that can deal with changes pretty well, and they've built up systems to be able to handle that. But there are some that are just, they're just adapted and their whole system is set up to handle one set of conditions, and if you're outside that conditions, you're not in good shape. So it varies from organism to organism. Right, to summarize it all up, so thinking about life in deep ice, either on Earth or in the ocean worlds, we know that it is an active habitat on Earth. It is still relatively unexplored. We've only got a few really deep cores that we can go and extract and play with. It does seem to be a good analog to the ocean world icy crust. You have the temperature and the pressure conditions, low light, all that is all very similar between one place and another. So it's a very directly applicable analog. I'll also double down and say that it is going to be the first habitable environment that we will encounter as we go and drill down to the ocean of an ocean world. So it makes absolute sense to have some way of interrogating it and looking for any microbes that have concentrated and sat into those micro pockets. And we know now that life clumps, especially in ice, and so we need to have a way of detecting those hotspots. And with that, I will leave it all and kick it out and be able to answer any questions. And I do have a few backup slides if you all want some really hardcore stuff, especially talking about biotech. Anyway, go ahead.
Interesting to know that wasn't the hardcore stuff. Well, thanks, Mike. Just real quick, for those on the line, I'll mention that we'll continue taking questions, and people are free to stay on the line and chat. At noon, though, the recording will stop, and I actually have to run away for a minute. So you'll be left to your own devices at that point, Mike and Kevin. But if there are any questions for Mike, feel free to put them in the chat or unmute and ask them here.
Okay, I'll ask one. This is Rosalie. How about using this instrument for Enceladus? Maybe there will be material more deposited right on the ice that you wouldn't need to drill down?
Yeah, we actually did call that out in the paper, where the depositional environment at Summit Station in Greenland could be very similar to the ice small, tiny grain deposition that you have at Enceladus. And in fact, I'll even, just reiterate that the snowfall that you have, or the airfall that you have happening at Enceladus, that may actually be recording the entire, depositional pattern and organic microbial history of those plumes. So you might actually be able to learn a lot about the deep history of the plumes by, drilling very shallow and near some of the vents of Enceladus.
Yeah, great. That sounds really exciting.
Okay, going to the chat. Have you visited any other analogs for ocean worlds other than Greenland?
I have not, but I do know that some of the technology has been examined looking at the WAIS ice core from Antarctica, and they found very similar concentrations using sort of a similar instrument where they did map it out. So it appears that this whole life clumping thing and making these, millimeter-sized blobs does appear to be a thing, and we just have to figure out why that's happening.
So, can you talk about the growth rate of microbes in deep ice? Are they actively reproducing? Are they trapped there? What's happening?
So for the Greenland ice, where you have low temperature, it is preserved. I believe they have shown active metabolism. I'm trying to remember exactly how they did it. I want to say that it was through a labeling feeding study. Where you fed it, I believe it was like a stable isotope labeled glucose, or maybe it's radio-labeled glucose, and then looked at what happened next in terms of chasing down the isotopes or the radioactivity. But it is slow, as you'd expect. So at very low temperatures, your growth rate is going to be very slow, which means these culture experiments are going to be very hard to do as well.
Thanks so much, Mike. So we've exhausted questions in the chat. People are still free to ask voice questions, though. I'm going to stop the recording now because we're at the top of the hour, but I wanted to thank Mike and Kevin again, both for great talks, as well as the Network for Ocean Worlds co-leads. So Kevin gets thanked twice. And right, next quarter, we're going to be having our final episode of this first year looking at life in ocean worlds. There will be life on the surface and in atmospheres of both the Earth and the other ocean worlds. I'm really looking forward to it. It's probably the one I know the least about.